Actually, inside the cell itself there's lots of structures that we need to talk about, and these structures are otherwise known as organelles, and we're going to talk about a few of them. We're going to start with the nucleus, then we're going to talk about the ribosomes, then the endoplasmic reticulum, and the Golgi apparatus. So these are some of the structures within the cell itself that have very, very important functions, and we'll break them down one by one and just discuss their various functions, why they're there and what they do. So let's start by talking about the nucleus of the cell. And the nucleus has two main functions. One is to maintain the integrity of the genes, so maintain integrity of the genes within it. And the genes are obviously very involved in reproduction of the cell and reproduction of the human body as a whole. And the second activity, and this is equally as important, is it controls activities of the cell. So controls activities of the cell. So what's the structure of the nucleus? Well, there's a number of structures in the nucleus that we need to consider. First of all is the nuclear envelope. So the nuclear envelope is what's surrounding the nucleus. So this is a double membrane which encloses the nucleus, and it isolates the nucleus from the cytoplasm. So it's a double membrane. Isolating it from cytoplasm, which is that fluid which is inside the rest of the cell. We've also got the nucleoskeleton, which just acts as a support to the nucleus, and it's much like the cytoskeleton, which acts as a support to the cell as a whole. So that's just as the function of supporting the nucleus. And then there are within the membrane nuclear pores, and these nuclear pores allow the transport of molecules across the membrane. One of the things we need to talk about within the cell are the chromosomes, and these obviously play a very important part in the reproduction of the cell. And the chromosomes basically are a linear DNA which is organized into structures, and there's approximately within each cell 2 meters within each cell of DNA. So you can see there's a lot of that there, and normally it's stored as chromatin. And it's only during division that is actually formed chromosomes. And then finally there's a discrete density within the nucleus which is called the nucleolus. And this job is to synthesize RNA and assemble ribosomes, which we'll talk about in a moment. So this synthesizes RNA and also assembles ribosomes. Ribosomes are next, and ribosomes are the sites of protein synthesis. So and there's two types. There's free ribosomes, which, as you might expect, are kind of floating around, and these are concerned primarily with synthesizing proteins for use outside the cell. So proteins for outside the cell. And then there are those which are attached to the endoplasmic reticulum. And these ones are involved in synthesis of proteins destined for the cell membrane or for outside the cell. So for proteins in cell wall or outside the cell. Then we can talk about the endoplasmic reticulum. And there's two types of endoplasmic reticulum. We start with the rough endoplasmic reticulum, and it gets the name rough simply because it's studded with ribosomes. And the function of this is to synthesize proteins and it also acts as a storage area for newly synthesized molecules. So storage area, and along with the Golgi complex, this will also synthesize and package molecules, so it works along with the Golgi complex as well. To package synthesized molecules. And then, as I said, I don't know if I've got enough room, but as I said, you've also got smooth endoplasmic reticulum. And this is the site of fatty acid, phospholipid and steroid synthesis. So synthesis of fatty acid, phospholipid, which play a great part in the phospholipid, which play a great part in the cell walls, and steroid synthesis. And in muscle cells in particular, you get something called the sarcoplasmic reticulum, which is the muscle equivalent of the endoplasmic reticulum. And this is an area where calcium ions are released, and that triggers contraction. So in muscles, calcium ion release. So the next structure is the Golgi complex within the cell. So what happens is that the endoplasmic reticulum is responsible for synthesizing or making the proteins. These proteins are then moved into the Golgi complex, transported to the Golgi complex or apparatus to be processed. And the Golgi is made of several layers called cisterns. And the proteins move through these layers. So the proteins move through the cisterns, move through the layers, where they continue to be adapted and packaged. And they're packaged into vesicles. So this is like a membrane around them, if you like. And those vesicles can either transport to the cell wall, so they can go to the cell wall and be excreted in the cell wall. And examples of some of the proteins where this might occur are things like hormones and neurotransmitters. So they're transported to the cell wall, the vesicle bursts, if you like, and the proteins are released into whatever is outside the cell. Or they may stay within the cell, and an example of protein that stays within the cell is the lysosomes. And the job of the lysosomes is to break up macromolecules. So the bigger molecules that might have entered the cell, examples of these are the polysaccharides, so complex sugars, proteins, and lipids. So other proteins and some of the fat cells that come in as well. So that's what happens with the Golgi complex. So the proteins developed or synthesized in the endoplasmic reticulum, moved up to the Golgi complex, which move through the cisterns and the Golgi complex. They're adapted and changed depending what function they have once they leave, and then packaged into vesicles, which then go to the cell wall and excreted out or stay within the cell, for example, with lysosomes.